So let me first of all introduce my colleagues on this work. On the top right is Heike Reiser, who is also my wife and my boss indeed. And in the middle is Bettina Schlitt, who is the technician who has done all the molecular work that I will be talking about. So these are the good guys, and these are the bad guys. This is Arion vulgaris. It's a big slug. It's about that big. Formerly, it had been misidentified as Arion lusitanicus, so in the literature you might come across it as referred to by the name lusitanicus. And this is our compost heap, and you can see, first of all, that it can achieve quite high densities, and you also get these are beans here, you get a feeling for how big it can get, but it can get bigger than that. This shows you that it can be quite variable in colour, and this is a mating pair. So like most terrestrial slugs and snails, it's hermaphrodite. They exchange sperm reciprocally, but they can also sometimes use their own sperm. They're usually reluctant to do that, but they can do. The colour is also variable in the sense that when they're young, they have a different colour. So this is a juvenile slug. So this time of year they might still be this colour, and then they will develop a more typical adult coloration. They are an annual species, so they become adult in August about, and will be laying eggs in August and September. And then all the adults will die off by October or so, and the next generation will be born, will hatch from the eggs partly before the new year and partly in the springtime. I'm afraid, bad news is, you've already got it in Hungary. So there's this excellent new guide to the slugs of Hungary, and one of the authors is over there. And this is the distribution of Arion vulgaris in Hungary. You'll see that actually you're better off than some parts of Hungary. It seems that it really is less common in this part of Hungary, perhaps because it's drier. We don't really know. But it hasn't always been in Hungary, and this is a species that has spread across Europe over the last 75 years or so. So I'll come back to where it's actually native in a second. But the first records, clear records of it, are from England and Switzerland in the 1950s. And each panel here is a decade. And it's not shown up very well, the colour, but the magenta colour is if that country recorded that species in that decade. And the blue is if it was already recorded. So you can see how it's spread through each decade. So a few more spots in the 1960s, and then, as I said, Hungary, 1980s. 1990s already pretty widespread over Western Europe and some of Eastern Europe. In the 2000s, the former Soviet Union republics had been colonized. And the most recent dot here is Armenia. But in the meantime, it's also been recorded from Canada and Mexico, and most recently also from the east coast of Russia, so Sakhalin and places like that. I'm sure it will spread more widely globally. So where did it actually originate from? Well, there was a paper written in 2014 by some German molecular geneticists claiming that it's always been in Central Europe, and that it's native, and it's just that it became more common, and that's why people noticed it. And I very firmly believe that this claim that it was a misperceived invasion is itself misperceived. I don't believe that it was ever native in Germany or Central Europe. And I'll just present some evidence about that, and it's also genetic evidence. So here we have a gene network, and for anybody that this is not familiar to, each straight line represents a mutation to a single base pair, and each vertice here corresponds to a particular genetic haplotype of the standard barcoding mitochondrial gene, CO1. And at each of these vertices I put some coloured dots, and the coloured dots, one dot represents one locality, and the colour of the dot represents where we actually collected that slug, or where somebody else collected that slug. It's not all my data. One pattern you'll see immediately is that there's a great big star shape, and that is characteristic of a species that has rapidly expanded in the recent past, which we know is the case in this species. But it's and the other thing we can notice is that the brown dots here, which are Eastern Europe, which are clearly where it's been introduced, when you've got good historical data that it wasn't there and it is there. These brown dots are not just around this star shape. There's some dots up there. There's some brown dots here. So it seems that it's not. There's more to it than that expansion there. Okay, another way to look at it is to colour the network according to where on the network it is, and then plot these colours on the geographical map. And you'll see here that the black spots here, which are so distant to the others that we don't even connect them, they're at least 11 base pairs different. They turn up in the western part of the Pyrenees. And these green dots, which are a long way away from the centre here, but also not part of that radiation, they're also mostly in the Pyrenees. There are some, it doesn't show up very well, sorry, but there are some green dots here, which must be an introduction, and one green dot there. So it looks like the greatest diversity in terms of where on that diagram the haplotype occurs is in the Pyrenees. So we believe that the Pyrenees is the origin of this species. And there's some further information here. Incidence of private haplotypes. So the idea is that if they're evolving naturally, not being spread around by lorries and garden centres, that their ability at dispersal is really not very good. They will tend to remain in the same place, and that therefore different localities will have different haplotypes in them. And that means that you will find a high proportion of the haplotypes that you find only at one locality, and that's called a private haplotype. And if we compare here across the diagram, here's the Pyrenees here, and then there are various other regions here, and the proportion of haplotypes that are private is 40 and 55% in the Pyrenees and the middle of France, and tends to be much lower in the other places. It's only 13% in Germany. And another way of looking at the diversity is the probability that if you take two localities at random and you take one haplotype from each locality, what's the probability that those haplotypes will be the same? And it's much lower in the Pyrenees than in any of the other regions, only 0.03, and it's more than double everywhere else. And another argument is examining what's called the isolation by distance pattern. The idea is that if they're in their natural habitat and limited by natural dispersal, then by the time your descendants have travelled very far, it's likely that they will have mutated in some way, because dispersal is so slow and difficult. And therefore you will predict that in their native area, you get a relationship between geographical distance and genetic divergence. And you wouldn't expect that if they're being spread by lorries across Europe, okay? So what's it look like? Well, these are two measures of that correlation between genetic and geographic distance. And again, we see that it's much higher in the Pyrenees than in any of the other regions that we've examined. So, yeah, we think it's native in the Pyrenees. Okay, so I've told you about its origin and I've told you about how quickly it spread across Europe, but we became interested in looking at the finer scale of invasion. Yeah? And we did that by looking at the slugs in our particular area. And we work in this town of Görlitz, which is on the German-Polish border. It's a very nice old town if you're ever passing through. It's worth a stop. It was not destroyed in the war in any way, so it has all the old buildings. This is a view of Görlitz, actually with the river Neisse running through it, and that is the German-Polish border, so this picture is actually taken from Poland. And what's special about our work is that we have successive collections over several years monitoring the spread of this species. In fact, we've got over 3,500 animals that we've examined, and it may not sound too impressive if you think you can just look at an animal and say, "Oh, that's the species." But in fact, there's a native species that you cannot distinguish on external characters. You have to dissect them. So over 3,500 animals have been dissected to identify them. I'll talk about the characters later. So this is a map of Görlitz. This is the river Neisse running through it. This is Poland. This is the German side, and there's a scale there, so it's 3 kilometers. And we first found Arion vulgaris in Görlitz in 1994, and these are these 2 magenta spots here. And I will also plot yellow spots here. These are occurrences of the native species, and they marked places where we only found the native species. So the yellow dots indicate that probably Arion vulgaris is not there yet. Okay, so we noted it that year. There was a paper published, not by me, but 4 years later we noticed it in a couple of other places. Yeah? And where it occurred before, I've marked in blue, so you can see what are the new places and what are the old places. Again, some areas where we only found the native species. The native species is called Arion arta, which I might refer to later. Okay, so we got interested and we started more serious collecting. So we sent students out to collect slugs. Each dot here is a whole collection of slugs. It might be 10, might be 20, might only be 3, but yeah. What you can see is that there are many areas where in 2000, the year 2000, that seem not to have been colonized by Arion vulgaris yet, but the species clearly had reached all 3 corners of the town. Yeah? It's spread over the whole town, but not everywhere. That's the next year, 2001. That's actually the first occurrence in this part of Poland, but we weren't really searching in Poland, so it's incidental. Again, some areas without vulgaris, but always more common. 2002, well, really, it is almost everywhere. There's an area here where it's clearly still absent from. So good collections of arta, but we didn't find any vulgaris. The next year, 2004, yes, it's really becoming quite rare. And it's also reached this area here, which is an old extinct volcano with native woodland on it. So it's also invading native habitats. 2005, it's clearly absent from that area which was holding out for arta. The only yellow dot here is in Poland. So you've got a feeling from how long it actually it takes for the species to colonize a town, sort of 10, 12 years, something like that. So it's an extensive and very fast invasion. And what I haven't been able to say so far is that it not only occurs where the native species occurs, it actually drives the native species extinct. So we can't find the native species anymore. But in that period when they both co-occur, it hybridizes, and that's what I'm going to talk about now. Oops. I'm not getting a response.
Maybe button.
Okay. I told you I'd talk about the identification characters. This is the reproductive anatomy of Arion vulgaris, and the characteristic, the character is this long cylindrical oviduct, and inside this oviduct is a kind of, I don't know, little plate, I guess. And this oviduct is everted during the copulation. So that's working again, that's good. So this is the oviduct of one species and this is the oviduct of the other. And they use this ligular structure to make contact with the partner. We don't know exactly what's going on, but in this species they remain in this posture for 4 hours typically, so they're doing something. Partly they're manufacturing a spermatophore which they exchange. And this is the other species, the native species. It actually has the derived condition. In that case, that they don't have this cylindrical structure. They have a more sort of rounded structure at the bottom of the oviduct, the outside of the oviduct. And inside it, the ligulus is sort of bent up like that, whereas not extended like that. There are some other characters about fineness of finger-like projections, but we have a suite of 5 characters we use. But the main one is the shape of the oviduct. And we made some plasticine models to try and understand the variation a little bit better. This is the vulgaris with this long cylindrical oviduct and the ligula here. And this is Arta, the native species, where it's more crunched up into a ball. And then we find these intermediate forms. And we have 3 classes of intermediates. We have the ones that look more like Arta, the ones that look more like vulgaris, and the ones that we couldn't say, somewhere in between. And these intermediate forms occurred between the years 2001 and 2007 in Görlitz. So when both species were present or one had just disappeared. Yeah? And because the invasion on the Polish side started a little bit later, we found the intermediate forms later on the Polish side of the river. So that's evidence, I think, quite good evidence already for hybridization. But we have some further evidence. One is that we put slugs together in the lab and observed their mating behaviour. So can the 2 species actually exchange sperm? And it's quite surprising that they can, because they have very different genitalia, as I just said, and the ligula is a different shape. There's further differences. One, vulgaris mates for 4 hours, and Arta mates for only 2 hours. And another difference is that vulgaris, the earliest part of mating, it tries to curl round the partner. In fact, they sort of spin round. And that doesn't happen in Arta. So when you put the 2 species together and they couple together, one is trying to rotate and the other is sort of puts its foot down. So you can see that they're in a rather sort of awkward, uncomfortable looking position. But nevertheless, they persist. And you can see here, just see the spermatophore actually being exchanged. The names are old, I'm afraid. This is vulgaris, which wants to continue another 2 hours, and this is the native species, Arta, crawling away and pulling out a spermatophore and leaving a spermatophore. So it is successfully exchanged. Is that not? It's about a third as successful as pure pairs of each species. So they have some difficulties, but they can do it. But to really prove hybridization, you need to have some nuclear markers, and we haven't got those, but fortunately somebody else developed them, a Swiss group with a PhD work of Jemenova, who I think is Czech. And she developed 16 microsatellite markers, and she's looked at the contact zone between Vulgaris and Arta along an altitudinal gradient in the Swiss Alps. And she showed that indeed there were genetic hybrids and that they backcrossed in both directions to the parents. Okay, so that's looking at probably a static contact zone between the two species. The sort of dynamic move-in contact zone that we have to deal with may be rather different from the point of view of population dynamics. Okay. No, it's gone too far. Here we are. There's some work by Currett and colleagues in 2008 that predicts massive introgression of a resident species genes into the invaders' genes if they have hybridization at the time when they coexist. And there's several nice arguments behind this. The first is that when the first colonists arrive at a site, it's likely to be a single colonist, and therefore it's kind of forced, if it's going to mate, it's forced to mate with one of the resident species. It's not quite so true in our case because they can self, but they're reluctant to self. So indeed, when we look in the field, we do find in these situations mixed pairs copulating with each other. That's half the story. The other part is that the animals that will colonize the next step of the invasion are quite likely to be the offspring of those hybrid, the result of that hybridization. One reason is that they're just in an adjacent spot. The contact zone is moving progressively, so it's quite likely that the virgin area will be colonized by a recently colonized area. And the other idea is that at that early stage of colonization, there isn't a very high population density of the invader, and therefore it's not going to suffer very much from intraspecific competition. So Currett et al. Modelled this, and they predicted this massive integration. And that argument applied just to neutral genes, but you can see that it might apply even more strongly if the native species had some useful character that would aid the spread of the invader. For instance, if it was in northern areas, if it was more resistant to cold weather or something like that. On the other hand, there might be selection against taking on the novel genitalia of the native species, and maybe against the mitochondria of the native species. So the characters we use typically to identify these species are the genitalia and the mitochondria. Yeah. So we might, because we're using those for identification, we might not notice all this introgression. So to test these ideas, we use the microsatellites that Jemenova and colleagues had developed. And I should perhaps say a little bit about microsatellites. They're fairly short stretches of DNA consisting of a series of repeats of simple repeats, really, of base pairs. And because of these repeats, during replication, they tend to mismatch. Yeah? So rather than being like that, they sort of slip a cog. And the result is that it's a fairly variable, the length of this segment is fairly variable, and therefore you just measure the length between the primers, and that gives you a measure of which allele you're dealing with. It's not ideal as a character. One thing is that you can easily get back mutations or convergent mutations, and the length of the microsatellite tends not to be characteristic of a particular population or even of a particular species. So you're actually looking for patterns of variation for a whole suite of microsatellites. And also, it's actually quite awkward to score because this mismatch during replication also happens in the test tube. So you get this, if this is length along here, you get a kind of stutter where it's the amplification of the gene has gone a little bit wrong and not been faithful. So you're sort of interpreting these patterns. This is one homozygote, this is another homozygote, and this is the heterozygote, yeah? It is a nice clean example, but they're not all like that. And so it involves sort of hours of looking at computer screens trying to interpret whether you've got a heterozygote or which one you've got. So what I would like to say about microsatellites is that at the best of times, they give you a kind of probabilistic estimate of the ancestry of that particular individual, and there is a sort of noise aspect to it. People often assume that molecular work is sort of all clean and gives you digital results, but actually it's a bit messy, more messy than you might imagine. Here are the results, and this is just for the animals that we looked at morphologically and were quite sure that they were one species or the other and not a hybrid. And these ones here are the native Arianata, and these ones are the vulgaris. And each thin column here corresponds to a single individual. And the more green there is, the more we estimate that the ancestry is of the native species, and the more purple the ancestry of the vulgaris, the invader. So for this example here, it's about sort of 3/4 native and 1/4 vulgaris. Perhaps it's an F2 individual as a result of hybridization a couple of generations before. And what you'll notice is that our anatomical identification is actually pretty good. There might be a couple of ones that are sort of hybrids, but a lot of this round here could just be noise and error. And the other thing we notice is they're actually arranged chronologically within each species. And you don't, for the vulgaris, you don't get more and more green during the process of the colonization. So there's no evidence from this that they're actually permanently, the vulgaris is taking on the genes of the native species. Okay, these are animals that we thought were hybrids on the basis of the anatomy. These were the ones that we thought were nearer to Arianata. These are the ones that we thought were nearer to Arian vulgaris, and the intermediates in the middle. And again, green means more like Arta. And there's a sort of a good rough agreement, I mean, at least a correlation between what the microsatellites suggest and what we found from the anatomy. There were about 4% of the sample were these potential hybrids. And what you can observe is that, you often do get mixed ancestry quite clearly. This is clearly 50-50, but there are sort of 3/4 and more dilute effects. This row here is the score from the barcoding gene. So it tells you about the ancestry on the mother's and grandmother's side, maternal grandmother's side. And what you'll notice here is that we do have some individuals that are mostly Arta in terms of their nuclear genome, but nevertheless they might have a vulgaris mitochondrion. And actually, the same is true in the other direction as well. We get both cases. So, both species can act as mothers of hybrids. Okay, so I've talked a little bit about interspecific hybridization, and the microsatellites also tell us something about intraspecific hybridization. So in the case of Arianata, that would be hybridization between the subspecies of Arianata. And I'm not going to talk very much about this at all, but just say that there are 3 subspecies of Arianata, which you can't identify from the colour, although there are trends. You can identify them on the basis of the mitochondria, the anatomy, and of course also on the basis of microsatellites. The agreement isn't perfect, but there is clearly some differentiation there. This is the distribution of the 3 different subspecies, and it happens that in Görlitz we have all 3 species there. And there's an interesting story about their distribution. It looks as though 1 or perhaps 2 of them of these subspecies are actually also invaders, but invaders from sort of 50 years ago or longer. But I haven't got a time to talk about that now. And anyway, we've got the problem that we can't study that anymore because they've all been driven extinct by the vulgaris. So we've only just reliant on material that we collected earlier to look at that. But there's also variation within vulgaris. So this is a kind of plot, first 2 principal components of the results of the microsatellites, 2 different types of analyses in the 2 different panels. And this is the variation of the subspecies of Arianata, but there's also this variation here within vulgaris in our particular area. Here's a plot of these 3 different, what do we call them, races, 3 different races of vulgaris. And you'll notice that the red ones kind of nearly all have a distinct distribution, that they occur in these 2 places, and they generally don't occur very much in the middle of the town. And then the blue one is also just sort of mostly centred on this park. What we think is, this is a bit of a guess, we think that the red is probably something that came from a different population. Perhaps was from a different town or a different garden centre, so that it sort of has a slightly different history. But we think it's possible that the yellow and the blue might have had the same history, and that the divergence is just a founder effect. Perhaps there was just one special, one slug happened to arrive in this park and spread from there, but its microsatellites are nevertheless unrepresentative of slugs that arrived elsewhere in the town. So it's just a hypothesis. We can't answer that, but but there are these 2 different possibilities. What's perhaps as interesting is if you look at these 2 sites, these 2 red sites here, and that's one column along here and one column along here. And again, each column is an individual. Each column is an individual. And what's interesting is that these yellow and blue microsatellite patterns only turn up very late on. I mean, it's been around for almost 20 years here before these types of vulgaris appear in the ancestry. So it looks like there's rather poor mixing, and I want to explore that a little bit further in a minute. To do that, we started a new study site, and it's not far from Görlitz, but it's on the Polish side. So that was invaded 10 years or so later. So we had the chance, we had the experience already of Görlitz, and we had the chance to start a new study in a sort of area where we expected the invasion to begin and to follow it all the way through. The place is called Görlitz. Yes, we could catch the start of the invasion because it's a very small area. We could actually establish which were the slugs that arrived first and genotype them. We studied them from 2013 to 2021, and every August or September, when there was a nice wet, damp evening, we would go to this site and we would find a slug and try and collect 20 slugs from its immediate neighbourhood, and then we would walk 10-15 metres further on and look for another slug and then collect another group of slugs. So we've got sort of different localities within the site. That's the site. There's the scale, so it's really quite a small site, 75 metres. That is all the sites that over that entire period. It just gives you an idea of where we were collecting from. I mean, we didn't collect here because we couldn't find any generally, but we tried to collect in roughly the same place each year. It's a sort of village green in the middle of a Polish village. Nothing very special. Sometimes it's grazed by horses. Sometimes it's mown once a year, something like that. There's a little stream running through it. And that is, in fact, where we found the first vulgaris specimens. So, this is going to be a succession of maps, and I must explain what they all show. This lower part here shows the results of our anatomical determination. And each dot is actually a pie diagram, and the size of the dot represents how many individuals we collected from that site. So a pure red dot would be the native species, Arian arta. A pure blue dot would be the invader, Arian vulgaris. And then ones that are green, which don't show up very well, they would be hybrids. Yeah? And you can see this was the area which I just showed the picture of, where we first found vulgaris. It's already very common here. We think we found a hybrid here. But here we've actually sort of examined it a bit more closely using the microsatellites. So for a sample of these animals, and it wasn't really a random sample, it was partly random, but sometimes slugs were particularly interesting to us, so we've selected them. So for instance, that hybrid there, we wanted to look at the genetics of that. We looked at the microsatellites, and so these dots here are the estimated ancestry of each of these individuals. One dot, one individual. And again, it's they are pie charts, but in that case it represents ancestry, and in here it represents the mixture of individuals. And you can see here that indeed around here we had two hybrid individuals, so probably the invasion had actually happened a year or two before that we got there. Vulgaris in this corner and a single vulgaris in that corner. And in between we only find arta. That's the next year. You can see again there's two distinct areas, vulgaris there, and some more hybrids, more green points here. Next year, 2015. Well, arta's almost disappeared by the looks of things. There's one sort of hybrid individual there. We think actually this is a bit of an artifact. It was a year when we collected rather late, and we have the impression that Arta dies out earlier in the autumn than vulgaris. Because we think it's an artifact, because the next year the Arta is back again. So it's probably just had disappeared in the previous year a bit before we collected. It was the latest collection of all the ones we made. Okay, so what's happening here? Well, now Arta is certainly in a minority, really. There's some hybrids here. Actually, yes, that was confirmed by the microsatellites. Okay, the next year, 2017, we collected a lot more there. Still, both species are persisting over this quite limited area. 2018, it was ever such a dry summer. We went there once. We've managed to find 2 slugs. So, yeah, sorry, that's the missing data. 2019, well, it seems to be mostly vulgaris. And 2020, the surprise is some pure Arta have turned up again. And that presumably is that they've spread from somewhere else. Yeah, so it's not all invasions in one direction, apparently, which is interesting. And I think this is the last year, but again, we had a single pure Arta that arose. But basically, the invasions happened, yeah? Okay, so let's look at that data a little bit more quantitatively. In this column here, each panel is a year, so the earlier events happening there and the later events down here. Along the horizontal axis, we've looked at the estimated proportion of the genome that is Arta. So we expect all the vulgaris to be here and all the Arta to be over here, and the height of the bar indicates the number of individuals in each case. There's just a histogram. So indeed, to begin with, we have a lot of vulgaris, and we have a lot of Arta, and we had a couple of intermediates, some hybrids. And by the end, we've just got a few hybrids that are sort of very close to vulgaris. And I think, the sample size is quite low, but you kind of get the feeling that there's a shift with time. The hybrids are getting more and more vulgaris-like, which is not a surprise, but it puts a sort of time scale on it. We also looked at the mitochondrial DNA of these hybrids, and they've been coded as red for Arta and blue for vulgaris. Now, it's difficult to see, but there are some red ones up here. But otherwise, all these hybrids have vulgaris mothers. Now, as I observed earlier, that's not true everywhere. You can have hybrids that have Arta mothers, but at this particular site, we only observe that. We don't know why. There might be some selection against Arta mitochondria in vulgaris cells. The divergence of the mitochondria is about 7%, so it wouldn't be surprising. But again, we don't observe that elsewhere. And it may just be that the vulgaris are much more successful, they lay more eggs than the Arta do, and that would also bias it towards more vulgaris mitochondria amongst the hybrids. So as a last point, more or less, we want again to look at variation within vulgaris, and this has worked out quite neatly. I'm going to show you the same diagrams again, just from the microsatellites, but in this case, we've marked 2 types of vulgaris using the microsatellites. There's the dark blue type and the light blue type. And you'll see just by chance the light blue type was there and the dark blue type was there. And the next year we get the same pattern, a couple of the light blue here There's actually a light blue here, but that didn't seem to have any progeny as far as we can see, because we don't find it there again. And the dark blue there. And next year, and now we've got a sort of, they've met, and we've got a offspring of these two types of vulgaris. And then the next year, so they're gradually expanding out from each direction. More mixture, progressively. Yeah. So that's the last year. So what's interesting is that even after 8 or so years, you can still see the signature of where the slugs originally were colonized. Yeah? So it's a contrast that the vulgaris has spread very quickly across the field side. But these two varieties have to be very slow to mix. I think the reason is this idea of the intraspecific competition. When there's only the native species there, that doesn't seem to be serious competition. They can spread through those populations. But when they meet another vulgaris population, yeah, their rate of reproduction is slower because of intraspecific competition, and that's why they don't meet. And actually, this goes back to some classic results from the sapir snails, which you might have heard of. So they're very highly polymorphic shells, so very easy to score. And what was discovered in the 1950s and 60s on grassland in Britain was that you got these so-called area effects, that you've got large areas of uniform habitat that were, say, all yellow or something like that. And then an adjacent area that looked environmentally almost the same had a banded shell, or a brown banded shell. And people were puzzled by this, and there were all sorts of elaborate models talking about the homogeneity of the gene pool, and that they can't, specialized races and things like that. But there's actually a much, much simpler explanation. After the First World War, sheep grazing decreased, and therefore the grassland became longer and became more suitable for these snails. And they colonized out from particular hedgerows where they'd survived. So they expanded rapidly over an area of nice long grassland. And then they came into contact with another colony, another expanded colony from somewhere else. And where they met, they didn't mix very well. Yeah? So you've got these apparently sharp boundaries just because of the intraspecific competition. Okay. So some take-home messages. Arion vulgaris can drive Arion arter extinct within about a decade. That's the sort of scale of the things. Arion arter does hybridize with the invader, the Arion vulgaris, but it looks like the genes are diluted away on present evidence. And then the last point, once Arion vulgaris is established, the population mixing between different groups of Arion vulgaris is surprisingly slow. We're not quite finished yet with this work. We're collaborating with a group in the Natural History Museum in Stuttgart, and they're about the ancestry. And some of the things they're showing are extremely interesting. Like, it seems like between these subspecies of Arion arter, there has been hybridization in the more distant past. And probably involving vulgaris as well. So it's quite a complicated story. And the other thing they can do is they can actually find particular areas of the genome. They can map particular areas of genome that do seem to have introgressed from the native species. And of course, it's very interesting to work out what genes are in that area. So there is a whole genome sequence for the big Arion. So there is a hope of actually discovering whether they're actually taken on particular useful genes from the native species. Okay, I think that's about time to finish, so yeah, I'll stop there.
Thank you very much. Questions? Questions?
Thank you very much. Very interesting story. For me, there was a missing puzzle or something that I didn't understand. So, what is the ecological secret to the success of their invasion? And so, if they're so successful and they can invade so rapidly, what prevented them from doing that earlier? You know, so if they're in the Pyrenees, they could have spread a long time ago. So what's
I think it's a very good question, and I think the answer is we don't know. I mean, people have found ecological differences, but whether they're the significant ones or not, it's extremely difficult to work out. I guess you could say, why didn't it happen earlier? Maybe there is more transport going on of goods, and it's also happening more quickly, so that things don't die en route. And that's, of course, a global phenomenon, not just with this species, that things are moving around the world much more quickly and much more extensively. So that's the best guess, but it could, of course, be that some magic mutation happened that suddenly enabled it to happen. But there's no evidence for that.
Yeah.
Yeah, thank you. Are the invasive species more likely to self-reproduce than the native ones?
Probably not. They seem quite reluctant to self-reproduce. There are estimates of it, but I don't know of good evidence in the native species. So, but yeah, what happens if you keep them in captivity? They're kind of reluctant to produce eggs, but eventually, if they haven't got a partner, they do lay eggs, and they are fertile. So it's a kind of last resort to self. That seems to be the. We've got lots of paternity experiments in other groups of slugs, but not so many not really in this species, because they're quite awkward to keep. They take a whole year to develop, and they don't seem to mature very easily in captivity. And they're big things, so you need a big space to keep them. So we tend to do all our paternity experiments and selfing data on other smaller species. And so probably the best estimate comes from looking at the genetics, and you can estimate selfing rates from homozygosity and things like that. Evidence suggests that both species can, but not very often.
Just a general question on hybridization. How would you define a hybrid? What portion of the genome would have to come from a different species to consider it a hybrid?
Yes. Yeah.
And
do you see it? Yeah.
So I'm just curious, and in terms of the conservation status of hybrids, how would you, in this case it's clear, but what if it's two protected species, one is highly protected, maybe it shows up as a hybrid in another protected species, and you would have to apply different management strategies to that.
Yeah.
So how would conservationists treat hybrids? These are just general questions. I'm just curious about based on this.
I think there are real cases of this where we've only got hybrids left, and what do you do with it? I mean, you can try and back cross in the right direction to enrichen it, and of course, some people are just satisfied with getting the appearance of the animal right. These white wolves that people have evolved in. That's just the external appearance that you can get right. It's a problem, isn't it? You can't easily purify them unless you can. Perhaps one day we can do it when we can routinely sequence the whole genome and get it a little bit more precise. Yeah, I know you can work. I mean, we know that we've all got a bit of Neanderthal in us. And then the question is, how much of the entire Neanderthal genome is there in different people? If you could breed it back together again, how much would you get? And it's not an entire Neanderthal, it's sort of, I think, 30% or something. It's surprisingly high, but yeah.
Other questions? Yeah.
Thank you so much. Do these organisms pose a problem to the ecology? Do we have to worry about?
Do they cause a problem to agriculture?
No, to any ecological conditions. I mean, do they have any negative consequences to the ecology?
If you've got a garden, I mean, you really notice when this species arrives. It's you suddenly can't grow things that you could grow. So that's one thing. But they have a negative effect, of course, on the native species, but they also have ecological effects as well. So in some areas in Sweden, for instance, the ground flora has just disappeared. Yeah? So the native ground flora in native habitat is no longer there, which is probably a massive effect. And not only on the ground flora, but on all the things that eat it. And there's also, there's some papers on some mosses and other lower plants that depend on dispersal by slugs, and these big slugs tend to digest the spores more than native slugs. So yeah, I think there are bound to be lots of considerable ecological problems, and actually, I mean, just when you go for a walk in the evening, you tread on the things. You know, it's just unpleasant. Yeah.